I'm Darsh Mandera, and welcome to our second workshop for YBS 2022. Thank you all for being here. We're really excited to get this opportunity to share our knowledge as well as maybe learn a little bit from you guys. And yeah, so workshop on computational methods in biological research. So, like Toba, I'd also like to start with a quote. This is He's the winner of the Turing Award for Computer Science, which is often called the Nobel Prize in Computer Science. And he said that biology and computer science are related. I'm that at their interface, great discoveries await those who seek them. And that's why I'm so excited to get to introduce a couple computational methods to you, data frames and classifiers, and also give you guys the opportunity to come up with your own CS/bio scientific to draw on what you're learning from my workshop, as well as what we learned from Toba's workshop earlier today. We have 4 key goals for this workshop. A little bit about the history of computer science techniques in biology to you. And then as a group, we'll get to work hands-on with data frames in Python. And don't worry if you don't really have any experience with Python, I'm gonna walk you through everything. And also don't worry about having like an IDE open or installed. There's some available online that we are going to use as a group to work with Python. So we're going to get to work together hands-on in Python, which is a key technique for biological research. And I'll also, later on in this workshop, I'll explain what data frames are, as well as classifiers and how they're important in biological research. You'll also get to learn in computational biology and how these techniques are being used in them, as well as get the chance to come up with your own scientific questions at the end. So I'll get started with a brief history of computational biology. Computational biology is a really young and emerging field, but its origins actually go all the way to the 1950s, when taxonomists first started using computer science to classify organisms. If you're not really familiar with taxonomy is really just organizing organisms into different groups like species, genre, clades, kingdoms, domains, etc. And so in the 1950s, using computer science methods to efficiently and more accurately organize organisms into different groups. In the 60s, these same techniques that taxonomists use were extended to analyze the DNA of organisms. And by the 1980s, computation really shifted towards using artificial intelligence to make predictions. This is super important because now in computational biology, what we're really looking at large-scale biological data and using that to make predictions about how something will behave or what the best way to approach something will be. Details on this briefly. So what emerged in the 1980s, this emergence of AI to solve computational biology, a huge focus of computational biology today. And this has been bolstered by the Human Genome Project, which has curated a ton of data on the human genome by these artificial intelligence methods to make prediction, because AI is all about analyzing data, and the more data you have, the better predictions you can make. So the Human Genome Project has been really fundamental to computational biology, especially for studying humans. I'll talk a little bit about research, and my own research is actually based on these last 2 points, these last 2 steps of the history of computational biology. It's all about AI and the Human Genome Project. For the past few years, I've been working on using cancer patient data, particularly their genetic mutations and microRNA alterations, to predict personalized targeting with AI. Through this research, I've relied heavily on data frames and classifiers, 2 methods that we'll learn about today that will allow us to work with biological data. Now, I guess I'll spend another minute or so talking a little bit more about my research in detail, and that'll help you get an understanding of what kind of are being asked in computational biology and how we're approaching them. And then we'll also look at some published papers and other research outside of my very niche area, broader sense of that. So I guess a little bit of context about cancer. Cancer is an extremely heterogeneous disease, very different from person to person, as well as different parts of the same tumor are genetically very different as well. And so the current approach is really an approach where patients receive the same treatment based on their type and stage of cancer. But this doesn't really account for how different cancer patient. And so instead, what we can do is focus on personalized medicine or targeted treatment, where we look at a patient's personal biological data that to predict the right treatment for them. And we can use this based on different biomarkers, which indicate which are things that indicate the presence of a disease. And biomarker would be a patient's genetic mutations or their microRNA alterations. Now, I guess that sets the biological and clinical context for about predicting the right targeted treatment for a cancer patient. And from a computational perspective, what I've done is I've looked at data from the Cancer Genome Atlas, which has patient genetic and microRNA data, and used data frames to work with this data directly in computer science programs, and then to make predictions about which drug is going to be best for a cancer patient. Now, I guess we can start working with data. I'll introduce data frames, and in a few minutes, we're going to actually get the opportunity to work hands-on with these. Data frames allow us to organize data into a table directly in our programs, and that's why they're so great for computational biology. We can take biological data and embed it directly into a however we want from a computational perspective. Today, we're going to be doing three different things with data frames. We'll create them, we'll join them together, and then well. And don't worry if you don't know what these three things are. I mean, the creating one's pretty self-explanatory, but the other two, if you don't know what these are, that's what this workshop is, what they are, what the benefits of those two methods are, and then we'll actually get to implement them as a group. So to get started, go ahead and visit this website, and I'll walk you through everything. So we're going to be doing a lot of this stuff together. We're going to go to online-python. And it may already have some stuff written. So just go ahead and delete that. I'll give you guys like a minute or so to get set up with this. We just want a blank screen here. Now, the first couple lines of code we're going to write are the following. We're going to say import NP and import, make a new line and write import pandas as PD. And then go ahead and run this code just because it's going to things, and this will have us set up for the rest of the stuff we're going to do. Now, NumPy and pandas are both libraries of Python. What this means is specialized, they're like specialized libraries and sets of code that are not necessarily part of the regular vanilla, but it allows us to focus in on a particular field of programming. And both of these really focus on data science, and they allow to work with different structures that don't really exist in basic vanilla Python. For example, data frames. And so, yeah, so these two libraries are work with data. So going back to the presentation, once you have these two lines of code set up, we're going to get into our first exercise. This exercise is based on just sort of an example research question. Let's say we want to research the correlation of patient protein consumption and height. There may or may not be some relation between these two factors, and let's say we want to study that. And a great way to study things like this is to use data and put them in a table, which we will do using data. So we're going to go ahead and create a data frame that looks something like this. We're going to have patients A, B, and C as protein in grams they're eating per day, and their height in centimeters. So together, we're going to go ahead and create in Python using these two libraries. So the first step to creating a pandas data frame that looks like that table we just saw is to actually put the data together in what we call an array using NumPy. An array is sort of like a base, and a data frame, we will then put that array into a data frame, which gives us a lot more functionality. So we're going to go ahead and create go ahead and create our first NumPy array, which we'll just call data1. So we write data1 equals NP, and then we will create our data frame within this, or excuse me, we will create our array within this. Creating the array is really just of the table. So we'll create our first row first. So going back to this, our first row, don't headers yet, those will come later. Our first row of data is 60, 65, and 52. So we'll go ahead and create that, 60, 65, and we'll follow that up with the second row of data, the patient height, which is 182, 1, and 170. So that you guys can get caught up with this. Now we can go ahead and actually put this data into a data frame. We will create, we can just call it DF1. It's easier to refer to things if we have short names because you don't have to type out the whole thing later. And to create this pandas or pd.data frame, and we'll put our NumPy array data1 into that data frame. And then now here's the part where we add additional information and actually construct those column headers and the row headers. So we'll say columns equal, and we'll just put the names of the columns in. So we have patient A, patient B, and patient C. So we can go ahead and add patient A, patient B, and patient C. So now we also need to add the row titles. So we'll say rows index equals whatever our rows were. So protein G and height CM. Now we actually have this data frame constructed. Go ahead and actually take a look at what this looks like. We just have to print it out. So we'll say print df1, and when you run this, sorry guys, this is supposed to say NP. That allows us to use that abbreviation. When we run this, we can go ahead and actually look at what this data frame looks like. So now, as you can see, we've actually constructed that data frame, that table that we saw in our slides. We have constructed it in Python. What this is going to allow us to do is it's going to allow us to work with it directly in the program and do all sorts of functions on it. I'll give you guys a moment in case you have some errors that, I faced an error myself because of that little mistake up there. So in case something's going wrong, I'll give you guys 2 minutes. I'll set a 2-minute timer so you can compare your code with mine and make sure everything's working just fine. And sorry, I don't want to be, if you guys have any errors right now, just use these couple minutes to think about how we constructed this, because the next stage is going to be you guys creating your own data frames on your own. And then we'll again regroup and we'll go over it together. But based on what you learned here, we're gonna create a set which you guys will get to do on your own. Okay, awesome. So yeah, so going back here, now we're going to get to create a second data frame with patients D. So I'll leave this up on the screen. I'm not going to switch over to the code, but based on the code that we just wrote for that first data frame, go ahead and try to construct this second data frame. I'll give you guys like 3 or 4-ish minutes to get this done, and then we can regroup and do that as a group. So based on what we just learned for that first data frame, here's a second data frame that we can try to construct, and then we'll actually get the opportunity to work with both of these together. Toba, feel free to unmute and let me know if he's like facing errors or something. And everybody, if you guys are like facing errors or something, or if you have any questions, go ahead and put something in the chat or the Q&A, and then Toba, feel free to let me know if anybody says something. Okay, so wherever you are right now, wherever you're at, we can go ahead and walk through this together as a group. Um, so the first step to creating this new data frame that we're trying to create is to create the forward. So we'll make a second data variable, data2. We'll say, and we'll create a NumPy array. We'll put in data, which is 62, 69, and 58. And then we'll create our second row of data, 180, 189, and 179. Now we can go ahead and actually construct our data frame, which we'll call df2, and we'll construct a pandas data frame. This time, remember to use the second NumPy array, so we'll say data2 names, say, patient D, and patient F. And then once we're done with that, we can go ahead and index names for our row names, which are the same as before, protein in grams, as well as the patient's height in centimeters. So now when we want to actually view this, we just switch out this print statement to print our second 2, We can run it, and we should be able to see our second data frame. There we go. Yeah, so this is the that we saw here. We have now constructed within Python using pandas and NumPy. So I'll give you guys a few seconds to catch up in case you missed something. And the other cool thing about Python is you don't actually have to online. So if we do this, we can actually see everything better. Um, yeah. So go ahead and wrap that up. And yeah, I'll give you guys a few more seconds to get caught up before we move on to the next task of our workshop. Okay, so now that we've actually created two separate data frames, is join them together. Now, from a biological perspective, there's different reasons we might want to do this. First of all, if we have sources and we want to combine them together, we may very well have data from two different sources that we want to combine together. And we create two data frames from those sources, and then we join them together so we can actually look at this as one data set. So now what we'll do, now that we've created these two different data frames, merge these together into one large data frame, like you see here. Now, this is actually surprisingly simple. We'll head over to the code, and it's really just one line of code that allows us to do this. We'll create a new joined data frame, and all we have to do is say df1.join df2. This simple method allows us to do a lot from a biological perspective. Like I said, we may very well have data from two different sources. And I know in my own research, I have had two different sets of data for two different cancer types. We can join these together to create one large data frame to work with. Yeah, so once you've written that line of code, we can go ahead and print out the join data frame. And what we'll see is exactly what we had in that slide. It's with all six patients A through F, along with their corresponding data in terms of their protein and their height. So once we've done that, there's one more I'd like to introduce to you regarding data frames. It's transposing data frames. So what transposing a data frame means is you're really flipping it horizontally, or you're switching the X and the Y axis. What this allows us to do is it allows us to look at data from a different perspective, the same data from a different perspective. When we make protein and height our columns and the patients the rows, it's just a different perspective on this data, and it can be really helpful figuring out what exactly you want to focus on. Or another thing you can another use of transposing data frames is that we may very well another set of data that we want to join our data with. And that's the same data, but in a different order. So by transposing it, we can make this data compatible with other. Now, transposing data frames is also really simple in Pandas. They've offered an amazing tool to do that. All you do is put in we'll just do it together. So we'll do a transposed data frame. All we have to do is take the data transpose, which in this case is join data frame, and put in no, it's not really even a word, just write .t after that. And when we do that, that pretty much just flips that data frame on its side and transposes it. So now we can print out that transposed data frame. And as you can see, the data frame has been transposed. And we have it on the other side, and we can see the same as what we have over here. I'll give you guys like a minute to wrap this up. And yeah, and feel free to let me know if you had questions about anything regarding data frames. And then we'll move on to our next topic, which is classifiers. Okay, awesome. So now that we've worked with data frames and done a bunch of different things with them, I think it's talk about um, how we can actually use this data to to make to actions in computational biology. So I'd like to talk about classifiers. Classifiers are a technique in artificial intelligence that looks at data and organizes it into classes. As you can see in this graph, we have two main groups, the yellow triangles and the blue circles. A classifier would draw a line between them and separate them into two distinct groups. What makes classifiers so amazing is that we can use them to make predictions. For example, in my research that I talked about earlier about predicting a cancer patient based on their genetic mutations and microRNA alterations, I use classifiers to look at a patient's genome and then classify them into groups where a group represents a targeted drug that they should receive. So classifiers lot of different computational biology problems, whether that's targeted cancer drug prediction, like my research, as well as other major research questions like Alzheimer's diagnosis, ecological analysis, or even COVID diagnosis. So computers huge impact in biology. Computer science has led to advancements in biological research in a lot of different areas. For example, scientists have mapped the 3D structure of proteins and genomes using computational techniques. They've also created accurate models of the human brain using computers. And they've also mapped large scale biological systems using these computational techniques. And all of these things have really benefited of computer science, including the methods like data frames and classifiers that you learned about today, because these large scale problems really aren't easy to do manually, and computer science allows us to do that a lot more efficiently, effectively, and accurately. The future of computer science is full of hope, and the truth is that all of you are the future of computational biology. While scientists have made great advancements in biological research through the application of computer science, there's still major challenges that remain unsolved today. I myself have been working in cancer treatment, but there's so many more problems that still need to be addressed. There's early detection of cancer. How can we apply computer science techniques to figure out when somebody has cancer as early as possible? This can allow us to treat cancer, which is a lot more effective than in its later stages. So as you can see, computational biology problems have some major implications and impact in a similar vein, can we detect Alzheimer's before it becomes apparent? Can we do this through genetic analysis, or are there other things to consider? While the Human Genome Project has done a great job of sequencing the human genome, can we also apply computational methods to sequence the genome of these are just a few of the problems that remain unsolved. And the great thing about computer science and biology is that you can pursue any problem. So, with that in mind, I'd like to take a brief 5-minute pause for you to brainstorm and come up with your own question. Based on what you learned in this workshop, data frames and classifiers, how you can really embed biological data into a computer science program, as well as use classifiers to make predictions, what you learned from Toba's workshop earlier about coming up with scientific questions. Try to think for a few minutes and see if you have any ideas about anything that you're passionate about, where you computational methods to a biological problem. We'll take a break for a few minutes while you guys think about that, and then we'll regroup and these ideas as a team. And also, just a little note, your the with doesn't necessarily have to involve the things we learned today, but it can draw from that as well if you want to. Okay, so it's been 4 minutes. Feel free to keep brainstorming if you want. I'm gonna actually sharing right now, and we can go and discuss any ideas that you guys had. So it looks like there's actually a question here in the chat. Someone's asking, what are some other common ways that data frames are manipulated? Actually a great question. I mean, we learned about creation, transposition, as well as joining. There's other ways of combining data frames together. We will join, but we can also merge data frames and we can join them vertically as well. So where you have columns that are the same and you're adding more rows to that data frame. Some other ways are actually splitting them up, so doing the opposite of joining them. We can by looking at the index of a data. Hey, look, I have this data frame that has 100 columns. I want that data frame from column 0 to 50. Or maybe we want to take different sections of that that aren't necessarily continuous. We can look at we can say, hey, I want to take this data frame from 0 to 50, 25 to 100, and then we can join those two together. Um, yeah. So another way is also to just there's actually a function just called dot drops. You can say you can have a data frame and say dot drop, and you can drop columns, whatever columns you want. So if you had 100 rows in your data frame and you wanted to drop columns 5 to 10, you could just say data frame dot drop 5. That would allow you to drop those columns. Another question I'm seeing is the pandas GUI compatible? Sorry, I'm not mean by that. Could you elaborate? We'll do Q&A at the end actually, so we can always come back to that. Now, if anybody come up with any research questions or anything that you're passionate about, go ahead and drop that in the chat.
Some people left research topics in the Q&A.
Oh, oh, I see that. Okay, thanks. Immunoinformatics, this is from Daniel. Immunoinformatics design to infectious disease. Yeah, that's actually super cool. I have a friend that's actually working, or I don't think he's still working on it, but he was working on this on something really similar last. So that's a huge thing in bioinformatics, vaccine design as well as immunoinformatics. That's super cool. Zero rare diseases. Also, that's not something I've heard about. So that's also super cool. yeah. So if anybody has ideas, feel free to leave that in the chat and or the Q&A, and I will start presenting again and we'll wrap up. Okay, so now that you've had the opportunity about data frames as well as a little bit about classifiers and how they're being used in research and come up with your own problems, I wanted to leave you with one last resource. And a huge thank you to Toba for sharing this with me. Um, Rosalind.info is a website that has a bunch of problems that that actually have a biological context, and they have a varying degree of difficulty. So we have you can well as some more complex ones. And you can this is a great way to get introduced to bioinformatics and computational methods in biology. Yeah, you can start with the easy problems and then work your way up to the more complex ones. So go ahead and if you want to go ahead and go to rosalind.info, there's a lot of that's a great resource to get started with problem solving in computational biology.